So what do you need to know here on antifungal agents? Obviously, I'm not going to ask you to draw any formulas. And just like with antibiotics, I want you to know the classes. Making sense? So, this. Polines. How they work? Disrupt the membrane. As usual, you don't need to know the names of individual drugs. Rhizofolvin. What's the mechanism? Inhibition of microtubule synthesis. Flucytosine. DNA and RNA synthesis are inhibited. And finally, azoles. Azoles are the best antifungals that we have. And they inhibit the synthesis of ergosterol, and ergosterol is the key component of the membranes in fungi. Make sense so far? All right. Now, the biggest challenge in treating fungal infections is this. Fungi are eukaryotes, and so are we, right? So trying to get decent specific toxicity can be challenging. So now let's chat about some mycoses. So we're going to start with cutaneous mycosis. Here's what you need to know. There are 3 most common genera that cause a variety of fungal infections on the skin, right? Then there's a candida, the yeast, that can cause fungal infections of the nails and various mucous membranes. We're going to stay some time and talk about this. Then there's Aspergillus. You see the statement, it's usually secondary. What does that mean is that somebody has a primary Aspergillus infection, let's say in the lungs, and fungi traveled somewhere through the bloodstream and erupted through the skin. I would say that for that particular patient, the skin, the cutaneous aspergillosis, is the least of the problems at this point. Okay? Am I making sense? Now, I want you to think about something. You may have had this experience before. You probably know someone who had one of these conditions. Okay? Making sense? You probably know someone with athlete's foot or ringworm. You probably know someone, maybe some kids, who had thrush. Okay? Or vaginal candidiasis. What's common between the conditions listed in this table and all others that I mentioned, like thrush and vaginal candidiasis, okay? They are on the surface. Why this is important? Why people who are otherwise healthy do not usually develop fungal infections? And the answer is temperature. Contrary to a popular belief, fungi do not like really warm temperatures. They prefer comfortable ambient one, like 24, 25 Celsius, like 78, 76, 79. Making sense to you? Good? Okay. So this is basically the shell temperature of the human body, the temperature in the skin, and the temperature on the external mucosa, like oral mucosa or vaginal mucosa. If a fungus gets kind of deep, gets inside, it's too hot, and there's fully functional immune system. So, why do some people still get those systemic mycoses, like meningitis or fungal pneumonia? Fungal infections become systemic in immunocompromise. Good? Everybody, am I making sense? Yeah, so that's the idea. Somebody's immunocompromised. There's going to be a problem with the fungus. Go ahead.
There's a range of medicines, like certain asthma medicines have a little bit of steroid in them. Like you
Okay, yeah.
Would somebody have something like that because they're immunocompromised?
Not enough, no. No, when I say immunocompromised, I'm talking about people with AIDS. I'm talking about people who are on immunosuppression due to the bone marrow transplant or receive some serious immunosuppressive medicines for autoimmune treatment. No, that is enough. Okay, now let's take a look at this, folks. So first of all, when we look at systemic mycosis, and remember, it's people who are immunocompromised. The portal of entry, by and large, respiratory system. Okay? Practically all of them are transmitted by respiratory route. So the first one is Cryptococcus neoformans. Okay? So it's a respiratory infection, I mentioned that. And then it leads to meningitis. Typical symptoms of meningitis are headaches, nausea, stiff neck. It's a hallmark disease of AIDS patients. What does that mean? It means that in most AIDS patients, if you have someone who goes to the hospital and they're diagnosed with AIDS, they're probably going to have fungal meningitis caused by Cryptococcus neoformans. Okay? So where does it come from? Birds. And what makes it even more notorious than just any regular garden variety fungus is the capsule. Okay? They encapsulate it, so it's really hard to get rid of them. Okay? There actually was an episode, I think, in House MD, loosely based on the case when they had a patient with AIDS, fungal pneumonia, and turns out this fella was like breeding pigeons and got exposed to the poop and got infected. Another one that causes meningitis, Coccidioides immitis, southwestern United States. It's very, very, very contagious, extremely contagious. Okay. Same story. You have respiratory infection first, meningitis second. Okay. For the reason unbeknownst to me, actually, it's classified as select agent. Just FYI, the term select agent refers to a potential bioterrorism threat. Okay. The research with select agents and the research with biosafety level 3 and 4, agents are under control of 2 institutions, and you can only imagine how great it is to deal with so much bureaucracy. Select agents is the prerogative of CDC. Biosafety levels is a prerogative of, surprisingly, USDA, United States Department of Agriculture. And certain violations of handling those agents are felonies, which is not stimulating intellectually when you do that research. Okay, now respiratory mycosis. There are 3 of them. You see this is a very short one. So you have pneumocystis, another hallmark disease for the AIDS patients. It's a fungal pneumonia. Okay? That make sense? Question?
No.
Okay. Typical pneumonia, so shortness of breath, cough, chest tightness, can be treated with antifungals, but the biggest challenge of treating someone who is immunocompromised is that there's no help from immune system. So for AIDS patients, it's actually doable because you can start the anti-HIV treatment. White blood cells will bounce back very quickly, actually, that happens over like a week or something, and then you introduce antifungals or whatever you need to use to treat the disease. Okay. Aspergillus. This is a fantastic disease, not in a good way. So this is pulmonary aspergillosis. Aspergillus, you saw it 100%. If you have moldy bread, that's aspergillus. Okay? Good? Now imagine somebody inhales it, and it starts growing like mold in the lungs. It's like you have a bowl of hypha, bowl of this fibers. Okay, I believe the first sort of case of pulmonary aspergillosis was discovered in turkeys. I mean, in birds, not in the country. Okay? There was a very enigmatic and cryptic epidemic of some unknown disease in turkeys somewhere in Europe in the late 19th century. There were corpses of these birds along the railroad tracks. So scientists started cutting them open. Turns out turkeys would pick up scraps of food that was thrown by passengers of the train on the gravel. That food would get covered with aspergillus. They would consume enormous amount of aspergillus. It would grow in their lungs and suffocate. Okay. Now, this is rare. Pulmonary aspergillosis is very rare. And blastomyces, it's kind of a two-in-one. So there can be skin manifestations, or in immunocompromised, it can progress to a pulmonary disease. Now, a lot of ideas in these lectures have to be reviewed by me, but I want to kind of digress a little bit and talk about the term self-limiting disease. You probably have heard that expression. Self-limiting disease, like, you get sick and eventually it goes away. I really like the statement that one physician made. Technically, every disease is self-limiting. Some of them are limited by death. So technically, he's right. Does that make sense? Not every patient recovers. Now, that concludes Unit 5. Again, I want to remind that it's a lot of memorization, but I can tell you that there's going to be 45 questions on the exam, but it will be graded out of 40. Do you understand what I mean? So 5 questions are extra. Okay? I despise that particular chapter. So on that note, we're going to jump to viruses and chat about these guys. Depending on timing, we may be able to get through some of the viruses. We'll see. Okay, so what are the viruses? I insist on calling them microorganisms. A lot of people don't agree with me. They are acellular and therefore not considered living. So if you have a question on some kind of nursing exam, are viruses living? The answer is no. My personal thoughts are irrelevant. The concept of viral species, as you can imagine, is weird, because when we talk about species, we talk about reproductive isolations. Why cats and dogs are different species? Because you can't cross them, and even if you could, the offspring wouldn't be viable, and even if it would be viable, it wouldn't produce viable offspring. It wouldn't be fertile. Okay, viruses don't even have cells, so the term species is usually used in the context of genetic similarity. Okay, I believe right now it's roughly like 270 viruses that actually cause human disease. Okay, so they're not particularly. Most viruses are absolutely benign. Now, very foundational feature of every virus is that it is obligate intracellular parasite. It cannot reproduce outside of the cell. The term survival isn't really applicable to viruses because they're not alive. So viruses get quickly inactivated outside of the cell. They lose the potential to infect the cell. Why do they need cell? They don't have ribosomes, so they can't independently synthesize proteins. And they do not have independent systems to generate ATP. They rely on the cell as the source of ATP. Good? Okay. Structure can be divided into two categories. So naked viruses, okay, contain two key components. They have a capsid, which is made out of protein. They have genome. We can talk about viral genomes a little later. There's enveloped viruses. So these guys, like HIV, they have the capsid, they have the envelope, and the envelope consists of proteins and phospholipids, and the genome, of course. So please know the composition of capsid and the envelope. One of my favorite questions, just by looking at this illustration, which these two types do you think will be more stable, more resilient, naked or enveloped? So the answer is naked viruses are more resilient, although it sounds counterintuitive. Here's why. A virus needs everything to infect the cell. Making sense? If it's an enveloped virus, it needs its envelope. But if you look at the composition, envelope contains a lot of phospholipids, which are very fragile. They really fall apart quickly. So if you have an envelope virus and just kind of you spray it on the table, in 4 or 5 days it's going to be gone. Because phospholipids just going to degrade. Making sense? Naked virus? All naked viruses are much more stable, much more resilient. Sounds good? Discovered, I mentioned that before, it's tobacco mosaic virus. He managed to filter it through the special filter, and it went through, so he called it initially filtrable agent. It's kind of interesting. The term virus originally was used by Pasteur to, he assigned it to any germs. And then viruses were like shifted to these structures and bacteria and so on and so forth. Size-wise, super small. Polio is not the smallest. So for the most part, you need electron microscope, right, to see viruses. But there are viruses that are absolutely fascinating, like Pandora virus that infect amoeba. Okay, Pandora virus has a size of 700 nanometers, which is 2 times more than the size of Mycoplasma cell, which is bacteria. And the genome size is 2.5 million bases, which is 5 times greater than Mycoplasma, and yet it can't reproduce independently. Okay? Classification-wise, one time I did the explanation of the Baltimore system. It took me an hour. I'm not doing that anymore. Okay? So basically, what I want you to know, viruses aren't living. If you would look at any living organism on Earth, living organism that consists of cells, all of them are going to have double-stranded DNA as a genome. Well, viruses aren't living. They don't have to. You can have double-stranded DNA as a genome, like herpes virus, is single-stranded, like parvo. you're going to have RNA genome in the virus, double-stranded, real rota, positive and negative single-stranded. So we're going to talk about positive single-stranded and negative single-stranded, what it means. So let's take a look. If you have positive single-stranded RNA molecule, you can immediately translate it into a protein. Okay? What if you have negative single-stranded RNA molecule? Are you familiar with film photography? Yeah? The film that you get out of the camera is negative. Everything is flipped. Same idea. When you have negative RNA, you need to convert it to positive RNA. There's no term for this process, but sort of the jargon. I'm going to put it in parentheses because it's not really transcription. You see what I'm trying to say? Like, it's not really double-stranded DNA into RNA, but you kind of get this mRNA that can be used for the protein synthesis. Does that make sense, the difference between the two? So negative-stranded RNA viruses have to convert the negative-stranded RNA to positive-stranded RNA following the complementarity principle. Now, capsid structures in viruses can be very different, and I can't stop marvel at the creativity of scientific naming. A lot of viruses have helical capsids, when individual proteins called capsomeres are arranged in a helix. The majority of, at least human viruses, have what's called icosahedral capsid. I have very limited ability to do the 3D imaging in my head. I can tell you that it's a 3D object with 20 sides, and each side is a triangle, if it helps. I personally think about completely different. Think about soccer ball. It's a decent approximation. It's not correct, but it's a decent approximation. If the shape of a capsid doesn't fit into anything, we call it a complex capsid. That's very creative. Okay, like smallpox or rabies or Ebola, Marburg viruses, they have complex capsids. Separately are phages. Now, phages, bacteriophages, infect exclusively prokaryotes, either bacteria or archaea. And this is specifically a so-called T-even phage. So they really look like alien spaceships. There's a capsid with a genome. And there's a tube covered with a tube sheath. And at the end, stale fibers. They are used, as you can see, to attach to the surface of a bacterial cell. And these are tail pins. They are used by bacteriophage to quite literally drill through the envelope of bacteria, cell wall and cell membrane, which is fantastic. Okay? So first, we're going to discuss the bacteriophage infection, and then we're going to talk about animal viruses. So bacteriophage infection can be either lytic or lysogenic. So let's talk about the lytic cycle and its 5 stages. So first stage is the attachment. This is when virus lands on the surface of the cell. Okay? Does anyone remember what any virus needs to get into the cell? The R word. Receptor. Yes. So bacteriophages aren't exception. I can tell what exception is. Plant viruses. Plant viruses enter differently, usually not through the receptor. So now that phage has attached, its genome penetrates and gets into the cell. You can see from this scheme that when phage infection happens, capsid doesn't get in, only the genome. Am I making sense right now? Once the genome is inside, it hacks the cell, cellular pathway, cellular resources, and starts the biosynthesis of its own genes. And this biosynthesis results in the production of a bunch of new virus particles, phage particles. Proteins are produced, DNA or RNA is copied, and particles are generated. And after maturation, the virus escapes the cell, resulting in the lysis. So cell is getting destroyed. That happens very quickly. So lytic phages, they just get in, reproduce, and kill the cell. Not immediately, but the fastest can do it in 30 minutes. Okay? Sounds good? Then there's lysogenic cycle. We talked about it before. We talked about both cycles actually in the context of transduction. So phages that cause lysogenic infection are often called temperate phages. When they get in, after the DNA penetrates into the cell, DNA is incorporated into bacterial chromosome, just stays there in the form of prophage. This is fantastic. It's a great advantage. Every time cell divides, it copies the prophage. So the virus spreads throughout population of bacteria. When bacterial cells become stressed, usually it is nutritional deprivation. Virus, like, you think of the virus like some person who claims to be your friend and hangs out in your basement, but when you experience financial struggles, they're like, I'm out of here, okay? And they destroy your house on top of that. So the virus leaves, gets out of the chromosome, and enters the sort of, this is why we call them lysogenic. They can result in lysis. Does that make sense? They don't have to, but at some point they will, but not immediately. So they do incorporate the genome in the chromosome and may eventually kill cell. Is it clear? You should be able to define the differences between the two. Okay, now some fun stuff. This, antibacterial therapy. Think about this. There were no new antibiotics discovered that would go into clinical application for the last roughly 20 years. Okay, it's a lot of time. And more bacteria become totally drug resistant. What you going to do? Well, one of the ideas is to use phages because they can kill bacteria, right? Like, the answer is on the surface. There are few teething problems. One of them, FDA really wants things to be standardized. And it's really hard to standardize the phage. You understand what I'm saying? It replicates. There can be mutations. You cannot guarantee that every phage is the same. You can guarantee that every molecule of ibuprofen is the same. That's the whole deal, you know. It's not a huge deal, but it is sort of a problem. Now the problem, if you design a phage that kills one specific type of bacteria, like species, like Staph aureus, can you guarantee that this phage does not affect anything beneficial? You can't. Bacteria can develop resistance. They have immunological mechanisms to protect themselves from phages. Do you follow? And finally, not a small deal, if you think about the structure of a phage, it's a lot of protein. So let me ask you this. Proteins that compose the phage, are they foreign to humans? proteins that compose a phage, are they foreign immunologically to humans?
Maybe not.
Yes, they are. They're not human proteins. They should serve as antigens, right? So if you take phage and you pump it into the blood for some reason, you may expect your immune system to detect it and produce antibodies. You're not going to get sick from it. Just the immune system will recognize and make antibodies. You can't exclude that possibility. And it may complicate the repeated administration because you have an immunity. Okay?
But aren't there millions of phages already?
There are. They mostly in the gut. Yeah. So, anyway, now the real clinical case that actually happened in the United States. So a couple of professors from UCSD, University of California, San Diego, they went for a vacation in Southeast Asia, I don't remember where a husband got infected with Acinetobacter baumannii. It's a pretty nasty bacteria, and when they arrived back to States, he was not in great shape, got in the hospital, and he developed systemic disease and sepsis. Basically had like oozing, purulent like lesions that pus was coming out. And he was in a coma, and was like, yeah, this dude's gonna die. Because it turns out that Acinetobacter was totally drug resistant. There's nothing to treat it with. He did not respond to antibiotics. So his wife researched options and found that at the University of Texas in Austin, there's a lab that does phage therapy research. So she got in touch, and they ringed FDA. FDA said, fine, this dude is not going to make it anyway. So they collaborated with another lab, actually from Navy, and in like 2 weeks, they got a huge amount of phage. People were working 24/7, literally. Like there were shifts. So they brought this phage to California and put the IV and hoped for the best. 3 weeks later, the guy walked out of the hospital. It was just amazing. There are some downsides that were detected. Indeed, immune response is generated. Repeated administrations may become less and less effective. But in many cases, it's the only treatment. There was a case of a girl with cystic fibrosis. She was in line for lung transplant, but she had bacterial infection in her respiratory system. And if you have infection, you cannot receive a lung transplant. So, this is going to be immunocompromised. And it was totally drug-resistant bacteria. And she received phage therapy, basically nebulizer with a phage, cleared everything. She got her lung transplant. So it's pretty amazing practice. Now, eukaryotic cells, stepwise similar. There are some differences though. So let's start with this. Let's start with attachment, same story. So for attachment, as you mentioned, we need a receptor. So receptor will determine the tropism. I think we used that word before. Tropism means where does a virus go? If there is no receptor, virus will not infect. Okay? So cells that do have receptor are susceptible. And cells that can support replication are called permissive. And you may wonder, why do we separate these things? Turns out, there are many instances when a cell has a receptor. So a virus can bind to this receptor. But it may not, even if it enters the cell, it may not be able to reproduce in it due to various reasons from immunological to biochemical. Or, there's a great example, mice don't have a receptor for poliovirus. If you will take a mouse and inject polio, like human poliovirus, it's not going to get sick. That's it. Stop. There's no receptor. You can take cells out of a mouse and, quote unquote, force poliovirus into the cell, bypassing the receptor entrance. Once it's in the cell, it will replicate in the mouse cell. So mouse cells, they aren't susceptible, but they are permissive. Does that make sense? Okay. Now, virus is getting in. You can see here that during the penetration of the animal virus, basically the entire virus goes in. It can be endocytosis, or the envelope can sometimes fuse, okay, to the membrane. But one way or another, the entire virus gets in. Now you need to release the genome. So this step that is not present in phage infection is uncoating, when capsid and whatever is left of the envelope falls off. Is that good? Then we get biosynthesis. Now, what do you need to know about biosynthesis? Mentioned that before. By and large, DNA viruses reproduce in the nucleus. Two reasons. One, DNA in the nucleus is normal, so cellular pattern recognition receptors will not see the DNA. So will, quote unquote, not know that it's infected. RNA viruses, for the most part, in the cytoplasm. Are there exceptions? Yes, influenza is an RNA virus. Biosynthesis is in the nucleus. Smallpox, double-stranded DNA virus. Smallpox is like a horrible, the worst thing that can happen to a cell. Gets into the cytoplasm, fully reproduces in the cytoplasm, kills the cell, leaves. Eventually, when during biosynthesis the genome is copied, the proteins are produced, you get assembly, you get mature particles inside the cell, and then they leave. Okay? So when these particles leave the cell, there is a difference between naked viruses and enveloped viruses. Usually, think about naked virus. It doesn't have to have an envelope, so it just pokes through, and the cell dies very quickly. It lyses the cell very quickly. Enveloped viruses, because remember, they have phospholipids. They steal these phospholipids from cellular membrane, and I usually give this comparison. Like when you watch the advertisement for chocolate-covered strawberries, and they like poke through the surface of chocolate, that surface merges behind them. So the cellular membrane can actually merge after enveloped virus steals the piece of it. Okay? Usually enveloped viruses don't kill the cell as fast. Still will, eventually. Okay? So that's release. Now, those key points that I mentioned are all listed here. Okay? Like, seriously, like all of it. And this is the viral growth curve in logarithmic representation. I just want to focus your attention. It's different from what you see for bacteria. So remember, in bacteria you have small lag phase, and then boom, you have logarithmic growth. You have eclipse phase in case of viruses. So this growth measures the amount of virus outside of the cell. When virus gets into the cell, there's nothing outside, and then you have the growth. Okay? Does that make sense? Cells produce more and more. What happens to the cells at the end? They die. Virus kills them, right? Eventually it kills all of them. And the growth curve flatlines. Because viruses cannot reproduce outside of the cell. Questions so far?
The number of viruses outside the cell?
you can take samples, and there's a virus titration. We don't talk about this. It's very, very technique specific. I just
Oh, okay.
no. If you like, unless you go into virology, you don't need to know how it's.
They saw it like under.
No. There's a for some virus, it's easier for some viruses and harder for others. But there are multiple ways to do that, and quantitatively, very quantitative. It's a large, enormous pain in the ass. But okay, fun stuff. So my plan is to talk about flu, polio, and kick you out of here. Okay? So, flu. Enveloped, which means it's not particularly stable. If you're a parent and you're concerned about the potential effectivity of toys that your child played with when they had a flu, collect those toys, throw them in the box in the storage like for 4 days. Virus will get inactivated. There are no cells. It's just those molecules just gonna degrade. Okay, it's gonna lose infectivity. That makes sense. Go ahead. Say it again? Pretty much. To some extent, yes. Bacteria would survive on a toothbrush. Viruses, not so much. Unless they're naked viruses, that's a different story. I can't imagine. But again, when you use toothbrush, you have to have viruses in your mouth. You have to transfer them on the toothbrush. And believe me, if you use your toothbrush with a toothpaste isn't great for viruses. They don't really survive that. That's a lot of detergent there. Okay? So there are two proteins that will sort of that are used in classification of influenza, hemagglutinin and neuraminidase. So functionally, this is the difference. Hemagglutinin, this blue stuff, binds to the receptors. Neuraminidase is the enzyme that basically controls the release of influenza viruses from the cell. So this types like H1N1 means hemagglutinin of the first type, neuraminidase of the first type. Okay, don't memorize the numbers or which ones are. Oh yeah, it's certain types of hemagglutinin, certain type of neuraminidase. It's the enzyme that's critical for the release of the virus from the cell. Now, epidemiology. Let's talk about this. First of all, it's zoonotic. It infects a lot of animals. If you're concerned about influenza in your pets, I presume that you have mammals, like dogs and cats. So they cannot be infected with human influenza virus, and reciprocally, we cannot be infected with cat or dog influenza. Okay? So the typical path for influenza virus to get to humans is this. It starts in the waterfowl or birds in general, and then it is passed to pigs. Pigs are the mandatory intermediate step, like you must have pigs. Okay? Here's an explanation why. So the influenza in birds is the GI disease. Okay? So they ship virus into pig enclosure. Pigs would snort it, and it's going to be upper respiratory tract disease in pigs. So virus adapts to upper respiratory tract and then transmit it from pigs to humans. Okay? Does that make sense? Now, you obviously can get the direct transmission from birds to humans. and that refers to bird flu. And I would like to explore with you in a dialogue fashion the difference between the regular, like human flu and bird flu. Okay? If you don't mind, I'm not going to talk about transmission of influenza. Everybody knows it's a respiratory, and the symptoms. Everybody knows what influenza looks like. Okay? We're not going to mention that. But virus needs a receptor. Okay? So, receptor in birds is alpha-2,3-sialic acid. Do not memorize that. So it's a certain molecule, okay? Receptor in humans, specifically in the upper respiratory tract, is different. Alpha-2,6. Can you see that? So birds, so GI. Turns out that the same receptor is found in the intestines and the digestive tract of birds we have in our lungs. So now I present you the scenario. You have someone who works at a chicken farm, and some of those chickens are infected with bird flu. It's a gastrointestinal infection. How does the virus get out of chickens? Feces, shit, yes. So this chicken's poop. This person works in that farm, right? And for some reason takes off personal protective equipment. And this person is tired. And this person takes a deep breath. And that aerosol, the virus aerosol, gets in the lungs. Remember, it cannot infect upper respiratory tract because the receptor doesn't match. So, but if it gets down in the lungs, the receptor for the bird flu is there. So the person develops viral pneumonia. This is why often bird flu is described as much more severe. Well, we don't really know. Okay, and this is why there were no recorded cases of human-to-human transmission of the avian flu. Because, let's say I have avian flu, and I cough, and I release virus particles, and you inhale them, just regular, like you normally breathe, okay? They end up on your upper respiratory tract. And what does bird flu find in your upper respiratory tract? Receptor that it cannot bind to. I mean, hypothetically, if somebody would sit down, open their mouth, and then another person with bird flu cough directly into their mouth while they take in a very deep breath, you probably can transmit it. I just find that kind of perversion very unlikely. Okay? So far, we don't have bird flu transmission. We'll see how it goes. Okay? So prevention. Hygiene. In Russia, there's a very strong belief that if you eat a lot of onion and garlic, it will prevent flu, which I think is absolutely scientific, because if you eat enough onion and garlic, people will just stop talking to you. It will stay away, and that will limit your chances of exposure. Now, let's talk about seasonal vaccines. They suck compared to other vaccines. And efficiency can be very low, and we need to explore why. And we are going to explore. RNA genome is segmented. There are 8 different segments of RNA. Am I making sense? So what does it create? It creates an opportunity for reassortment. There are 2 ways influenza can change over time. One of them is antigenic drift. This is the accumulation of mutations. This virus, when it reproduces, its RNA slowly accumulates mutations. Sounds fair? This happens all the time. And this is the gradual process. And then we have antigenic shift. So imagine medicine has 8 cards that are diamonds, and I have 8 cards that are spades. We represent 2 different flu viruses in the same cell. We take our cards, which represent genomes, put them together, shuffle the stack, and take 8 cards each again. We're both going to have diamonds and spades. This is a reassortment. In 2009, the infection of a pig with 1 pig virus and 2 bird viruses resulted in reassortment, and the new virus emerged out of that pig and caused flu pandemic. It wasn't any worse, folks. It was like a virus, I guess. Not any more dangerous than a regular seasonal flu. Just completely different. Nobody had any antibodies. So it swept throughout the world. Interestingly enough, that 2009 pandemic virus pushed out evolutionary the previous virus. So it replaced it. Viruses do it all the time. Okay, does that make sense? So we have 2 mechanisms, antigenic drift, which is slow, and antigenic shift, which is fast and abrupt. So why do vaccines suck? When virus accumulates mutations, it changes slightly the structure of hemagglutinin. And the vaccine from last year becomes less and less effective. Okay, and every year, this is what happens. A bunch of scientists, they get together, I think in Geneva, and basically they do an educated guess which circulating strain of influenza will be the most common. They publish it, and companies rush to produce vaccine based on that strain. If they guess wrong, we bone. Okay? And another problem with the flu, not a problem. So this is why, first of all, flu vaccine is not terribly efficient. And it is not aimed at preventing infection. Not a single vaccine is. They all are aimed at preventing disease. Okay? Like really bad disease. So if you look at efficiency of influenza vaccine in preventing people from dying, it's pretty good. Preventing people from being hospitalized, it's decent. Preventing people of simply getting the flu, not great. Okay? Does that make sense? So, again, I probably shouldn't say that, but technically, if you're a healthy adult and you're completely egotistical, you don't need flu vaccine. But there are some other considerations that you may think of. Vaccine reduces the chances to transmit the virus. Even if you're healthy adults, there's a site. It's called Families Fighting Flu. You can Google it. Site organized by people who lost their loved ones due to flu. They have personal stories. I read through some of them. 95% of them are healthy young people who were not vaccinated. Got influenza, died. So there's still a chance. And flu vaccine is extremely safe. Does that make sense? Treatment, it's not great. Okay? There was amantadine. Virus developed resistance. Then there was Tamiflu and Relenza. We're almost there in terms of the resistance. Now it's a new one called Xofluza. Don't remember what it inhibits. I think if you take it quickly, like first 24 hours after you have symptoms, you can shorten your disease by like 2 days. I mean, obviously, if you are an 80-year-old male with cardiac problems, you should get vaccinated, and even if you get flu, you should get a treatment, because influenza increases chances of dying from heart attack or stroke a lot. Okay, and in terms of the deaths, so like U.S. estimates that mortality fluctuates, like 40,000 people probably a year, but actual numbers are probably much higher because, again, that 80-year-old grandpa gets a flu and then gets a stroke. He died from a stroke, but the risk of having it, influenza increases by 60 to 70%. So maybe influenza. Making sense. Now, this is not, I always say that this is not an opinion, this is scientific fact. Flaviviruses are the best viruses. Okay. They're called flavi because flavus means yellow, yellow fever. And these are 5 flaviviruses I want you to be aware of. I challenge you to tell me which of these 5 viruses is not transmitted by mosquitoes. Tick-borne encephalitis, it has the word tick in it, right? Tick-borne encephalitis transmitted by ticks. All 4 others are transmitted by mosquitoes. To my knowledge, all flavies are transmitted by biological vectors, most of them mosquitoes. So all of them, all diseases listed here, are what's called biphasic disease. So, sorry, what does that mean? Okay. You see the statement, first phase is flu-like. You can read the virology textbook on each and every virus out of that list, and this is what it's going to say. First one is flu-like. My former boss, under whom I studied yellow fever, asked, so she talked to a guy who was in U.S. troops, deployed somewhere in Southeast Asia, and he had dengue fever, which is translated as break-bone fever. And she asked him, how was that flu-like episode? He said, and I quote, if I would be able to reach for a gun, I would kill myself. He wasn't physically able to move because every bone in the body hurts. It is like flu on steroids. Okay? It's really bad. Most of the people who get either of these diseases, they recover fully. It's super miserable, but they fully recover. It's bones, joints, muscles, everything hurts, head hurts, super high fever, but then you go back and everything's normal. In some folks, you get into the second phase. So, these two, dengue and yellow fever, hemorrhagic fever. Okay? Yellow fever just has a liver involvement. This is why people have jaundice. Should I say what symptoms are of encephalitis? It's brain infection. West Nile is also encephalitis. Okay? All good? Now, among flaviviruses, only these two are directly transmitted. So, see that? Specifically this cycle. So human gets bitten by a mosquito, mosquito carries it to Another human bites another human, they get an infection, and it goes on and on and on. This is why we call it an urban cycle. It is applied to yellow fever, it is applied to dengue. Sounds good? All right? Now, what about vaccines? I'm going to put an asterisk. These three, vaccine preventable. West Nile, nobody cares, because there aren't enough infections in humans in the first world countries. It's a problem in the developing countries, but they don't have money. There's a vaccine for horses, though. And dengue, there was a vaccine, and a lot of scientists were saying, dude, you should not deploy it. There are some complications that arose, and scientists were saying, you should probably wait. They deployed it and shut it down in like 6 months. Because I don't want to get into the details, but basically in some circumstances, vaccine was great and protective, but in a small cohort of people, it made the subsequent infection even worse. So they had to stop it. If you get into the second phase, the mortality is pretty high for all of them. And there is no specific treatment. Okay? That makes sense. Now, West Nile is transmitted from birds to humans by mosquitoes, Japanese encephalitis from rodents, and tick-borne encephalitis from rodents by ticks. But I want to focus on yellow fever and dengue specifically. Do we have, in the United States, do we have a transmission of yellow fever within the country, within the continental United States? We do not. The people in sub-Saharan Africa have a transmission of yellow fever. What's the difference? We have mosquitoes. We do. We do have mosquitoes, huh? Nope.
Is it the type of mosquito?
No. Same Anopheles mosquitoes. What? Okay, so are there mosquitoes outside now? No. When are mosquitoes outside? Summer. Can it be hot during summer day? Or even night? So when you come home and it's really hot outside, what do you do to be comfortable inside the house? Yeah, that's it. And if you don't have AC, what do you have to do? Open the windows and invite them over. Does that make sense? If you would look at the dynamics of malaria and other mosquito-transmitted diseases, they were the transmission was really low until like 1970s. And then we stopped using some chemical in 1970s. Anybody knows? You're very, very young. In 1970s, so there's a chemical, DDT. It's anti-mosquito chemical, super effective. Causes cancer in humans. It was like sprayed on the large areas, and it was not great. I mean, it was not great for mosquitoes, not great for humans as well. So they stopped using them, and the rates spiked. One of the ideas now, in terms of not spraying, mosquitoes that transmit malaria and yellow fever, they have a very interesting pattern of behavior. If you open the window, they will not just fly straight in, they're going to land at a windowsill. So one of the ideas is just to spray it with DDT, the windowsill, and leave it there. Mosquito lands, dies, that's it. Okay? Or increase the economic conditions, improve economic conditions, and so people can have air conditioning. Now, this, unfortunately, a heritage of 2015, when Zika virus outbreak was actually going on. It doesn't deserve that amount of attention. It's another flavivirus that's transmitted by mosquitoes. I'm going to skip all this epidemiology. I need to rewrite it. But it was interesting because there were interesting medical conditions associated with it. So the disease in adults, and even in kids, is very benign. So what you got rash, you got conjunctivitis, you got some pains. To my knowledge, there was one death. Some like very old immunocompromised person who died because he was just frail. But then at some point, scientists noticed that in kids born to mothers who experienced Zika virus infection during pregnancy, there's an abnormally high percent of microcephaly, reduced brain size. And this is the lifelong disability which affects both cognitive and motor skills. So I was glued to my headphones, listening to this, like how it all unraveled, and eventually scientists proved that yes, indeed, infection with Zika virus specifically doesn't necessarily lead to microcephaly in 100% cases, but increases the risk significantly. Now, this disease is not really super important because almost everybody who was supposed to be infected was infected. And people just have immunity and that's it. Okay? That's it. But at that point, people were freaking out. Like, can I go to Mexico for a vacation? Yes, of course you can. That's going to be the last thing to talk about, and then I'm going to let you out. Okay, so polio. I went berserk. Imagine that somebody asks a 9-year-old to write an essay, why do you like Captain America? And they just go on and on. That's me with viruses. So, of course, I'm not going to ask you about receptors and stuff like that. Some things that I want you to understand here, though. Single-stranded RNA virus. It is non-enveloped, which means it is resilient. Okay? Does that make sense? Infects exclusively humans, resides in water and soil, and can sit there active for months, up to a year. So infection is through the oral route. In 1940s and 1950s, polio was pretty rampant in the United States. What do you think was the most... What kids' activity was associated with polio the most? See it? Absolutely. Pools. Yes. Community pools would close. Okay? So, interestingly enough, when people get infected, the virus replicates in the gut. Oh, chlorine would not do much to this virus. It's that resilient. I mean, you could probably, if kids would agree to swim in bleach, sure, you know. So, and then it is excreted. But here's the thing. This neurotropic infection, the infection of the spinal cord, occurs in 1 out of 100 to 200 individuals. Because for a virus, it's a lousy perspective. Once the virus is in CNS, it can't go anywhere. That's it. It's a dead end for the virus. For really, that's it. Dead end. So, the virus spills over, gets into the nervous system, ends up in the anterior horns of the spinal cord. Anterior horns control your skeletal muscles. My typo. Flaccid paralysis. Muscle weakness, as you can see in these kids here on that photograph. They all have braces on the legs. Can you see that? It makes sense because if the virus spills over out of the gut, it probably is going to get into lumbar region of the spinal cord, right? And lumbar nerves, that's muscle control of the legs. But here, if it somehow manages to get to phrenic nerve, cause the diaphragm paralysis, breathing becomes a serious problem. I want to ask you a question. Can you breathe without diaphragm? Yes. You have accessory respiratory muscles. Well, first of all, you have intercostals, they're not accessory. And then you have scalenes, levator, scapulae. You can produce enough effort to inhale and exhale. But you have to focus on this. Because that automatic breathing is because of the diaphragm. So if you fall asleep, you're going to die. To help people who have this condition, iron lungs. Okay? To my knowledge, in the United States, there's one iron lung left. It's in the museum. The last person to have iron lung died a couple of years ago. He was a lawyer. That was like 63 or something. So he would sleep in the iron lung. But he would work like without it. Okay? So basically what it does, it expands your chest for you. And it was a big bummer in the beginning of COVID pandemic because people who required ventilation, what they did, they intubated them, right? So they forced air into the lungs. This is not how we breathe. That forceful delivery of air added additional damage to the lung tissue. So the idea was, what if we get iron lungs and put them there and we'll breathe for them? Well, we didn't have iron lungs. So. And we didn't have any company that could make it, or so basically. The vaccine was first. There is no treatment for polio. Okay? But there are a couple of vaccines which are phenomenal, and really interesting stories are behind them. So first is the Salk vaccine. It's inactivated. Get in a syringe, stick it in the shoulder. You're good. Okay? So what are the advantages? Injectable vaccine. Advantages, safe. I do know about Qatar accident. It's when a batch of vaccine actually contained active poliovirus and ended up in multiple people getting sick. But it was not a vaccine fault, it was a manufacturing fault. People did not do quality control. Okay? So it's safe, effective. It is, but there are disadvantages. First of all, it's a shot. Can I give somebody a vaccine shot legally? No. You have to have a medical professional, right? Second, it requires cold chain, which means you have to store it at a certain temperature. Okay? So, considering this, Sabin develops attenuated oral vaccine, and he and Salk become mortal enemies, because when he came to the US government said, why? We already have an activated vaccine. And it was the right thing to do. So Salk went to his buddy, Mikhail Chumakov, one of the leading virologists in USSR, and told him, dude, we have this vaccine, want to try? I mean, obviously not in these words, but you get the gist. And Chumakov said, sure. And they started vaccinating general population. Vaccine had very limited clinical data. So it was a huge gamble. And Chumakov realized that if it fails, he's not losing the job. He's going to a prison camp. Okay? it worked. They, oh shit, hold on a second. They practically vaccinated the entire population of the Soviet Union with the Sabin vaccine. There's one problem. So attenuated vaccine means that there are polioviruses inside. They can reproduce, but they're not going to cause the disease. However, those polioviruses in the gut can revert, go back to the wild type. The disease-causing ones. And in 1 out of 1,000,000 people who got vaccinated, one person developed vaccine-associated paralysis. And at the time, it was like, yeah, it's fine. Recently, there was an idea to develop a new vaccine. They got it down to 1 out of 10 million. Great, but not ideal. So, but the advantages, there are several advantages to this. First of all, it's much more tolerant to the storage conditions. Second, imagine I get vaccinated with attenuated vaccine, right? So the virus reproduces in my gut. I poop it out. I will poop revertants, but I will also poop vaccine virus. It goes in the groundwater, gets in the drinking water, somebody else drinks it, involuntarily, quote unquote, vaccinates themselves, which isn't a bad thing. And another thing that was very important, it's delivery. It's the oral vaccine. So a little sugar cube and a drop, literally a drop of vaccine. When my kids got vaccinated, they ditched the sugar cube. So just give it a drop, the person swallows, that's it. Can I do that? Probably can. So all these vaccination programs, like in Brazil, in India, in other Middle Asian countries, they were run by volunteers with no medical experience. Because all you need to do is just to give a drop. Okay? Make sense? So right now, United States use inactivated polio vaccine, which is expensive, and countries that have problems with the cold chain, with the vaccine delivery, most of them use oral polio vaccine. And finally this is the last thing we're going to talk about. So WHO constantly sets the goal to eradicate polio, which is ill-advised idea, because if you give people this live vaccine, they're going to shit the revertant. So we have to keep vaccinating people against polio, despite the fact that most countries eradicate polio completely. But you may ask, are there cases of polio in the United States? Yes, there were recently several cases in New Jersey. It was in a community, I think it's Pakistani community. So a lot of people are not vaccinated, and they acquired a revertant from probably somebody who was. So this is an old map. It pretty much screams update me. But if you look at this, these are vaccine-derived polio cases. What does that mean? It means that this country are diligently working on vaccinating the population, but some people are still not vaccinated, and they develop this vaccine-derived polio. This, though, kind of remains. It's a border between Pakistan and Afghanistan. Taliban. Dead serious. They had a vaccination program when volunteers went to villages and vaccinated kids. When Taliban took over, they started killing the volunteers. And people said, nope, we're not doing this. So there are like real wild polio cases, and that's the very last one. Okay? Any questions? Let's shut it down.